So this is a study with a whole different hypothesis and mechanism of action. So here we hypothesized that when we combine our regimen, high-dose regimen of vorinostat, Gemcitabine, Busulfan, and Melphalan, or GemBuMel, which causes massive DNA damage, with a drug which is Olaparib, which inhibits PARP. PARP is the first enzyme that intervenes in the process of DNA damage repair in the cells. So by inhibiting PARP, after causing massive DNA damage with our high-dose chemotherapy regimen, we hypothesized that the combination would be synergistic. So we did studies in our lab where we saw that in refractory T and B cell lymphoma lines, the combination of Olaparib with vorinostat, Gemcitabine, Busulfan, and Melphalan led to a marked increase of cytotoxicity and apoptosis. So in the clinical trial, we dose escalated Olaparib combined with vorinostat, GemBuMel, and we saw in 35 patients who had received a median 3 prior lines of therapy, where almost a third had failed previous CAR T. We saw that the regimen is, number one, safe, with an expected side effect profile of mucositis and clinically silent transaminitis, self-limited and reversible. And we saw that among 15 patients who had active disease coming into transplant at the time of enrollment, 5 of them in progression, active progressive disease at the time of enrollment, we saw 100% complete remissions. So all patients experienced a complete response, including patients who had failed CAR T and patients who were in progression at the time of transplant. And a median follow-up of 18 months and lead follow-up of in excess of 3 years, the event-free survival rate is almost 90%. So those are very encouraging results in a very hard-to-treat population, such as patients who fail CAR T or patients who have failed numerous chemotherapy lines.